Good morning, everyone. Thank you for joining and welcome to the Cynata Therapeutics Investor webinar. Presenting this morning will be CEO Dr. Kilian Kelly and Chief Business Officer Dr. Mathias Kroll, who will detail the clinical trial milestones of the past 12 months and the partnership strategy, and then take questions. If you would like to ask a question, please use the Q&A button at the bottom of your screen. You can submit them at any time, but we will be addressing them at the end of the call. And just as a reminder, you would have seen this as you were joining, but this call is being recorded, so please be respectful, and it will become available on the company website in coming days. I will now hand over to Chairman Dr. Geoff Brooke to formally welcome you all.
Thank you, Lauren, and good morning, everybody. Thank you for attending our quarterly update with this webinar. As you can see, Kilian Kelly, our CEO, is with us, as well as our new CBO, Chief Business Officer, Mathias Kroll. It's a very exciting time for the company. In fact, it's probably the most exciting time so far for the company. We are in the midst of 4 clinical trials, as I'm sure all of you are aware. We have our phase 3 osteoarthritis trial awaiting data. Our 1B DFU study also awaiting output of data later this year. We've commenced recruiting for our 2B GVHD study, and we have our renal transplant study underway as well, just awaiting our first patient. In addition, we've had another fantastic publication in arguably the most prestigious journal on the planet, follow-up data on our initial GVHD study, our long-term data now, which is just improving, getting better even still. We've acquired the TechSite wound dressing for the DFU application, which is a great advance, and the feedback from the physicians and from the marketplace that are actually using the product really like the dressing. So that's a big plus for us as well. We've added strength to the management team with Mathias joining us, and you'll hear directly from him, and you can make up in part your own mind when you hear him present. And so I'd just like to thank the staff who are working really hard to make Cynata the best that it can be. But most importantly, I'd like to thank the shareholders for their continued support. Without the shareholders, obviously, we'd be nowhere. And as I said in the opening, I think this is the most exciting time that the company's had. With that introduction, I'd like to hand it over to Kilian and Mathias to present for you now. Thanks.
Thanks very much, Geoff, and thanks also from me to everybody who's joined us today. We really appreciate so many people taking time out of their busy schedules to hear what we've got to say, and in particular, obviously, thanks to the continued support of our shareholders. So the presentation today will be reasonably brief because we want to leave enough time for the questions and answers session, but obviously we will cover the key points of our progress over the last year and where things now sit. So for those of you who are existing shareholders or who've been following the company for a while, you'll be well aware that the company is entirely focused on the development of a revolutionary stem cell manufacturing platform, which we call Cymerus. Now, this platform is being used to make what are called mesenchymal stem cells, or MSC-based therapies. These therapies have shown lots of potential to treat a wide range of illnesses, especially over the last 15 to 20 years. But the major obstacle to their commercialization has been that standard manufacturing methods require an ongoing supply of new donors. And that approach is associated with challenges with consistency, potency, and scale. And that's what we're trying to solve. We're trying to solve those challenges by using the Cymerus platform, which is based on another type of stem cell called induced pluripotent stem cells, or iPSCs. And this allows us to overcome that obstacle by producing an effectively limitless number of consistent and high quality MSC doses from a single blood donation. So it's a real game changer in how these therapies can be produced at scale. Now, we're not the only ones who've realized the potential in iPSCs. In fact, this field is really burgeoning around the world. There are lots of companies now pursuing iPSC-based therapies of different types, not MSC-based therapies. We are the only iPSC-based MSC clinical stage company at the moment, but other companies have seen similar potential in iPSCs to make other types of cells. Yet Cynata is still the leader of this field. We completed the first ever clinical trial with iPSCs worldwide. We've got orphan drug designation and a cleared IND from the US FDA. We've generated compelling clinical data in both graft versus host disease and diabetic foot ulcer trials. We now have 4 active clinical programs, including trials in phase 2 and phase 3. And we're going to have 3 randomized controlled clinical trial readouts coming up between late this year and early 2026. So as Geoff said, this is an incredibly exciting time for the company. We have a huge amount of really important news flow coming from our clinical trials in the relatively near future. Now, that's really going to build on lots of progress in the financial year that we recently completed. So I'm really pleased that we made material advances in all of our clinical trial programs. We completed patient enrollment in both the phase 3 osteoarthritis trial and the phase 1 DFU trial. We got the first patient enrolled in our phase 2 GVHD trial, and we got approval for our kidney transplant trial, which is now ready to go, and we're expecting the first patient very soon. We also released further encouraging clinical efficacy data. So in February, we released initial data from our DFU trial, which is really encouraging, and we'll show you what that is in a moment. And we also had additional data published in Nature Medicine from our phase 1 GVHD trial, which just further underlines the importance of that trial and how exciting those results are to the wider field. And as Geoff said, we've also strengthened our management team with the addition of Mathias. This is a new position, Chief Business Officer. The reason we created this position is really to help drive the next stage of the company's growth, especially with a focus on partnering and business development. And Mathias has an absolute wealth of experience in those areas, so I'm really confident that he's going to help us achieve our goals. Now, just to summarize our pipeline, the phase 2 GVHD trial, which is a Cynata-sponsored trial, recruitment and patient enrollment is ongoing. We're aiming to complete that by the end of this calendar year with results in the second half of next calendar year. The diabetic foot ulcer trial, patient enrollment is complete and results are not far away at all now. We're aiming to have them late this year or early next year. Then the 2 partnered programs, osteoarthritis, which is partnered with the University of Sydney and funded by the Aussie government. This is a phase 3 randomized controlled trial. Again, recruitment is complete and we expect results early in 2026. And then the kidney transplant trial, which is partnered with Leiden University Medical Center in the Netherlands. This one is approved and ready to go. We should have enrollment underway very soon. Patients are already being screened and so on. And the first cohort results should be available within a few months of the first patient coming in. Now, just to remind everybody about what our platform is and why we're doing this. So MSCs, as I mentioned at the start, have lots of potential for a wide range of medical conditions. And that's because they promote an immunomodulatory environment, which means they balance the immune system and they promote tissue repair and regeneration. So those properties have very wide applicability. But as I said, the main obstacle to commercializing these therapies has been the challenges with making these cells consistently and at scale. And that's what we can do. And this is where the iPSC part of the story comes in. So iPSCs are mature adult cells. So in our case, these cells came from a blood donation. And they're reprogrammed to become pluripotent. And what that means is they have the ability to reproduce themselves effectively limitlessly. So you can grow these cells in a lab and they just continue to produce more and more of themselves. They also have the ability to turn into any other adult cell type, including MSCs, which is what we're focusing on, but also they can be used to produce all sorts of other cells. They have similar properties to embryonic stem cells, but because they're derived from adult donors and not embryos, we avoid all of the ethical controversy that has gone along with embryonic stem cells. So we think that these are an ideal starting material for the scalable production of cellular therapies. Now, in terms of how our platform compares to a conventional process involves getting donated tissue, for example, bone marrow or adipose tissue, isolating the MSCs, and then growing those cells in a lab, which is a process known as culture expansion. Now, they need to go through a very extensive level of culture expansion, and the reason for that is quite simple. They start with a very small number of MSCs from each donation, but they need a pretty large number of MSCs to treat even one patient. And if they want to treat hundreds or thousands or tens of thousands of patients, which is exactly what we are trying to do, they need an even greater level of expansion. Now, that's a problem because MSCs undergo functional changes and loss of potency when they go through extensive culture expansion. So that means there's a limit to how much you can produce from each donor. So you have to go back and get new donors all the time. That's logistically challenging, but an even bigger problem is there's huge variability between MSCs derived from different donors, which means you get inconsistent activity. And that's an enormous problem because for a pharmaceutical grade product, you need to be able to show consistency from batch to batch and over time. So that's what we're aiming to solve with our Cymerus platform. By using iPSCs, we can make essentially all of the cells we'll ever need from a single cell bank that came from one donor one time. So that means we don't need new donors, so we don't have variability between donors. Then when we use our patented process to turn the iPSCs into MSCs, we don't have to go through a great deal of culture expansion at that stage because we can get our scale at the iPSC stage of the process. So we also avoid those problems like loss of potency as the MSCs are extensively expanded. So when you put all this together, we have a way of making cells with a high level of potency and consistency in a scalable manner. Now, moving on to our partnering strategy, I'd like to pass over to Mathias at this point to talk everybody through what we're currently doing and what we expect to happen over the next while.
Thank you, Kilian. Good morning. As you've just heard, Cynata has a great portfolio of preclinical and clinical indications. And following our second Nature Medicine paper, mentioned by our chairman, we've just seen another scientific paper come out where Monash University, the University of Melbourne, and the University of Manchester published on the effectiveness of our Cymerus MSCs in a model of pulmonary fibrosis. That's a great example of an indication which can, together with related indications, form a bundle or nucleus of a package which can be treated with Cymerus MSC products and be licensed or options out. These bundles are often defined by a route, dosage, number, and frequency. So as you've heard also, there's a very broad application potential of our Cymerus MSCs. And taking into account all of the different indications, there is a whole big universe of potential partners, ranging from multinational companies with a portfolio of therapies in large disease indications, hospital specialty pharmas, biotechs, device and advanced medical supplies developers such as in the field of wound care, healthcare providers, governmental organizations, and also other biotechs in stem cells which benefit from a source of highly consistent, scalable, and potent MSCs to form a credible proposition for their own technologies. All of these have different capabilities and requirements, and understanding these and developing the right story for each of them, that's our key focus. The business case for joint development and commercialization needs to be customized each time. The strengths and gaps vary across the value chain of the product. Next slide. And of course, we need to increase our activity at creating visibility, which is already very good. In order to ensure this very high level of visibility, awareness, and interest, it's important to be front of mind at the key partnering and scientific events that relate to our activities and engage in discussions with other companies and other organizations. For this reason, we present Cynata at key industry events, and you see here some selected ones, and this list is actually complemented by some smaller events. You will also see there's Biotech Showcase, JP Morgan Bio Week, and those active in the field know that this is a premier event where investments and partnership deals are made at C-level. We have also a good number of ongoing discussions, and new discussions are just starting at individual level, and that is very encouraging. So we are on a good path to prepare for deals, and you will certainly hear more of that in the future. Back to Kilian.
Thank you very much, Mathias. Just to remind everybody now of the great clinical data that we have already generated using our Somairus MSCs. Our first in human trial was conducted in steroid-resistant graft versus host disease, which is a really terrible condition, which is often fatal, and which at the time had no other treatment options that were approved. This trial has led to 2 publications in Nature Medicine, which, as Geoff said, is one of the most prestigious journals worldwide in our field. It's quite unusual for a phase 1 trial to be published in a journal like this, but the reason I think that happened is in part because it was the first ever iPSC completed clinical trial, but also because the results really were very encouraging in a condition where there is still a huge unmet need. Now, this slide just illustrates the results we saw in that trial in comparison to a phase 3 trial of a drug called ruxolitinib, which has recently been approved for the treatment of steroid-resistant acute graft versus host disease. Now, just to clarify, this was not a head-to-head trial. The green bars here show the results of Cynata's product from our trial, and the orange bar shows ruxolitinib in the phase 3 trial of that product. The blue bars are the control group in that trial. And what you can see is at day 28, the response rate with our product was a little bit better than ruxolitinib, but not dramatically so. But at later time points, what happens is the response rate with ruxolitinib tailed off, whereas the response rate with our product continued to improve. And perhaps even more importantly is the survival data. So the ruxolitinib trial showed that there wasn't really a survival difference between the active group and the control group at 18 months. In both cases, the survival was in the mid sort of 30% range, whereas we had 60% survival at that time point. And just to underline that, we still had a 60% survival rate at 24 months, but the 24-month survival rate in the ruxolitinib trial was not reported. It was stated to be not evaluable in that paper. So it does appear that we have not just good response rates, but also very encouraging survival rates. And we also have a very good safety profile with no safety concerns identified related to our product. And unfortunately, that is not true of ruxolitinib. Ruxolitinib is associated with numerous potentially serious adverse reactions. So overall, we believe that this product has the potential to really fulfill the unmet need in GVHD. Moving on to DFU, diabetic foot ulcers. So we have a novel product here. It's an MSC-seeded wound dressing, which we licensed the underlying technology for this product from TechSite Limited originally, and then very recently we announced that we've actually acquired that technology outright. In the ongoing trial, we released data from the first 8 patients per group after 10 weeks follow-up earlier this year. And we saw really encouraging outcomes. So a reduction in wound surface area, a median reduction of almost 88% in the active group compared to 51% in the control group. So we're obviously hoping to see a similar pattern when we see the full results from this trial, which, as I mentioned earlier, are really not far away now. It'll be late this year or early next year. And so just to summarize those catalysts, as I said right at the start, we have a huge amount of upcoming news flow from our clinical programs now. So the kidney transplant trial, we should see the first patient enrolled very soon and results of the initial cohort early in the new year. The phase 2 GVHD trial, we're aiming to complete enrollment by the end of this calendar year with results in the second half of next calendar year. The DFU results will be very soon, late this year or early next year. And then finally, the phase 3 osteoarthritis results should be available in the first half of 2026. So each of these trials is extremely important in its own right. Each of them has the potential to show that MSCs could fulfill a huge unmet need. So it's a really, really exciting time. Now, with that, I'll pause with the presentation there, and I think we'll move over to questions. If we have any, Lauren will read out whatever questions we've had submitted.
Yes, thank you, Kilian. We do have several questions here for you. We'll start with this one, which is regarding the recent news out of the Murdoch Children's Research Institute regarding lab-grown stem cells for bone marrow transplants. And somebody would like to know, what does this mean for Cynata and how will this impact you?
Right. Yeah. So look, that was a really exciting story that was reported in the last couple of days. It was published in another of the Nature journals and from a group here in Melbourne, which is great. They're obviously a very high profile and talented group. One interesting point is they're also using iPSCs, and it's another example of the huge potential that iPSCs have. And I really believe that iPSCs are going to be the way that cells are made across the board for therapeutic purposes. And the results they saw were quite exciting, but I think it's important to say it's very early stage. So it was in an initial study in a mouse model where they have made basically what are called hematopoietic stem cells or blood stem cells. And this is potentially a replacement for bone marrow transplantation in the future. But I think even the lead researcher on the project was quoted as saying that they are aiming to get into an initial human safety trial in about 5 years. So it would be fair to say that it would be many years after that before this product could be approved for human use. So it's a long way away, and obviously all sorts of things can happen over the course of a development program. So I think in the short term, it really doesn't have any direct impact on Cynata, but I think it's really exciting, and it sort of helps validate the point we've been making that iPSCs are really the way to make cell-based therapies.
Thank you. Now, the next question is regarding your DFU trial, and it is, could you please outline your proposed strategy and timeline for commercializing the DFU product, assuming the current phase 1 trial is successful?
Yeah, great question. So, we are obviously planning for success there, and we've been doing a lot of work on this, both internally and in terms of outreach to potential partners. I'll perhaps pass to Mathias to comment further on this, but in a nutshell, our aim is to partner that program, and I think with really positive results from this trial, we will be in a strong position to do that. Mathias, would you like to add anything?
Yeah, it's a product with really great potential. DFU obviously explodes in terms of epidemiology in lockstep with diabetes. We will see if GLP-1 agonists can actually make a dent in that. This is a great example of how large the space of our potential partners is, right? There are advanced wound care companies, device manufacturers, specialty hospital pharma, diabetes companies, healthcare providers, and so on and so on. And we've reached out and are starting discussions with some of these. Obviously, we've got a device that is feasible that can be further developed for industrial application. But of course, should one of the partners want to integrate this program into one of their existing technologies, we're also open to discussing that.
Thank you. I think that answers that question. Now, we have had several questions regarding your cash runway. We see with an expectation to take us through until halfway through 2025 calendar year. And there are questions around, will you be able to sustain the cash that you have for your current clinical trial pipeline? And are there any plans, or will there be plans for future capital raises?
Yeah, so obviously our cash position is something that we keep a very close eye on. As reported in our annual report at the end of June, we had about $6.2 million in cash. That is sufficient to keep us going right throughout this financial year and into the second half of next calendar year. And as explained there, the clinical trial readouts, the ones that are being funded by Cynata, are expected within that time frame. It is important, I think, to remind everybody as well that the kidney transplant trial and the osteoarthritis trial are not being funded by Cynata. So there is, they do not impact on our cash runway. And obviously we keep a very close eye on this, as I said. There are various sources of funding for the company. An important one is potential corporate partnerships, and we have brought some cash in the past through that route. Our objective is to do a whole lot more of that, and as Mathias has already outlined during the presentation, and I think in response to the last question, that really is a key focus now, and we have a lot of activity ongoing there. So that really is where we're focused at the moment.
Thank you. And to expand on that, could you please elaborate a little bit on the strategy to secure some of these partnerships or licensing deals? And are you able to touch on whether there are any conversations or negotiations in process for partnerships at the moment?
Well, I can certainly confirm that there are a number of conversations ongoing. Obviously, as I'm sure everybody will understand, we can't reveal any details of confidential discussions. In terms of the sort of broader strategy, again, maybe I'll ask Mathias to elaborate on that.
Yeah, probably that'll come back to some of the points I mentioned earlier, in that we obviously strive for very high visibility. So we go to different conferences. We make sure that we're visible to different functions in companies as well, scientific, R&D, regulatory, business, commercial. And we go to cell and gene therapy conferences, to International Society for Cell Therapy, to the general partnering conferences, the top ones being Bio International and JP Morgan Bio Week. We present at most of these conferences. And actually, again, very impressive how much endorsement we receive from the expert community in our field. So all of that builds, of course, to raise interest and to help us start new discussions. Again, Kilian, as you said, we obviously can't comment on precise details of where we stand with individual discussions, because we have to respect the confidentiality requirements of our discussion partners as well.
Thank you for that. I have a couple of questions now regarding the JFU trial. And the first one is whether you will announce the preliminary results after the 24-week follow-up period, or whether you will wait until the clinical study report is written at the end of the year or into early 2025.
Yeah, that's a very good question. So I suppose just to elaborate on the process, the final patient visit will actually be this month, and at that point, we need to make sure that all of the data is entered and what we call monitoring, which means it's all sort of verified and so on to make sure it's all correct. And then there's various data management activities that go on. And what that will lead to ultimately in a few months' time will be the database being locked and then the results being sent off for statistical analysis. After that, there will also be a formal clinical study report, but that will take some time. And yes, I can confirm that our intention would be to announce the results when we have the initial statistical analysis complete. At that point, we do know what the sort of headline results are. The report itself, for anybody who's familiar with a clinical study report, they're very long documents with a lot of information to go into them. So obviously that will take quite a bit of additional time, but we will be announcing the key results once we have them from the statistician.
Thank you. And the second question was, what was the rationale behind acquiring the DFU IP now versus paying milestones and royalties if the product is successful down the line?
Yeah, so look, I think first of all, it's simpler and better for us to have ownership of the IP, the intellectual property, rather than be the licensee of another small company, basically. So from a potential partnering perspective, rather than the partners having to then sort of do due diligence on the licensor, which was TechSite, it's all simpler. It's just us. They just need to deal with us. So that's one point. The other point is that, the cost of acquiring the technology was relatively modest at this stage, in that, as we reported, we were able to acquire that by issuing shares to the value of $230,000. So we believe that was a good deal for the company, and it puts us in a strong position to be able to offer this as a package. We now own the underlying dressing technology as well as the rights to the MSCs themselves. So it just puts us in a stronger position to go into those discussions.
I think just to complement, part of the acquisition deal, as Kilian said, at a very reasonable price, was also that there are some remaining success-driven milestones.
Mm.
Thank you. We'll answer a couple more. I'm very conscious of time, but people do appear to be sticking around, so they're willing to give us an extra 5 minutes or so. So this question, Kilian, is regarding the Medical Research Future Fund Cardiovascular Health Mission grant that was provided to Cynata in September 2022. And at the time, it was expected to run for a period of 2 years, which brings us through to now. When can we expect an update on this?
Yeah, so one small point I should correct is that the grant was actually awarded to our research partner, which was St. Vincent's Institute, rather than directly to Cynata. The project is ongoing and has generated a lot of data. We don't have a sort of a report to announce outcomes yet, but I expect we will be able to do that in the relatively near future. And this is another example of where we've been able to partner with leading academic researchers who can, who have access to non-dilutive funding, and we can really leverage the expertise not only of our partners, but also the funding that they can access. So this is another example of a project that really hasn't cost Cynata anything in terms of cash contribution.
And final question for today, which has come up a couple of times, and Mathias, this might be better answered by yourself. And this is whether there are non-disclosure agreements in place to facilitate some serious partnership and commercialization discussions at present.
Yeah, with all of these ongoing discussions, we currently have about half a dozen NDAs in place, and hopefully working up to more in the near future.
Thank you. Well, I believe that is all that we have time for. I appreciate everybody joining us this morning and giving us a few extra minutes. If we didn't get to your question, we do apologize, but please email it directly through to Kilian using the details at the bottom of the announcement or those that are on screen now. If you go to the final slide, the email address will be there, and we will endeavour to come back to you as soon as possible. Before we wrap up, Kilian, did you have any closing remarks?
I'd just like to thank everybody again for joining us today. It looks like we really had a great attendance, and as you said, some or most were able to even hang around for a few extra minutes. So we really appreciate all of the support and the engagement, and we look forward to really delivering against our objectives in the months and years to come. So thank you very much.
Thank you,
Thanks, everyone.
Kilian, Geoff, and Mathias. We will leave it there, and everybody have a lovely Thursday.